All right. Hi, everyone. Thank you for joining me today. My name is Jordan DeSantis. I am a clinical director here at ALS in North America. I oversee 3 of our North American clinical sites in Phoenix, Arizona, Torrance, California, and Purchase, New York. I have worked for the company now. It will be 13 years in December. And I currently have some direct oversight of our sunscreen and clinical product efficacy and claim substantiation testing. So today we are going to talk about sunscreen testing. We are ensuring safety and efficacy in a changing regulatory environment. Safety is at the core of everything that we do at ALS, so I thought it would be appropriate for us to start this webinar with a safety moment. Today, we're talking about sunscreen. So it is very significant that we are accurately and comprehensively testing sunscreen products. There are millions of cases of skin cancer globally. 90% of visible skin aging is caused by sun exposure. And daily sunscreen use can reduce the risk of developing melanoma, which is the deadliest form of skin cancer, by up to 50%. With this webinar, I'm hoping that we can discuss our rigorous testing methodologies so that we can help consumers protect themselves against these harmful UV rays, providing peace of mind for both consumers and us in the industry. First, I wanted to give you a brief overview, a little bit about ALS. ALS is Australian Laboratory Services. They have a number of different testing facilities in environmental, equipment reliability, food and beverage, mining and commodities, pharma, and for us, personal care and OTC. ALS has acquired a number of testing labs that some of you might be familiar with. Bioscreen here in Phoenix and in Torrance, California, Allergisa down in Brazil, IRSI, our New York location, Media Lab Science in Studio City, and most recently RCTS in Irving, Texas. We are now all one ALS under the ALS umbrella. Sunscreen testing specifically is conducted in 2 of our locations, our Phoenix, Arizona location and in our Sao Paulo in Brazil. Between these 2 sites, we are testing 75 plus sunscreen products per month. So our throughput, our capability is very good in order to get your products tested. So first, I thought we would talk a little bit about regulations. The first on the list is FDA. So this regulation is accepted strictly in the US. With the FDA regulations, we can determine your static SPF value, or that SPF number that's going on the label, the broad spectrum or the critical wavelength of the product, and if the product is water resistant. And water resistance can be claimed at either 40 or 80 minutes. The methods in this particular table are referencing the proposed sunscreen rule by the FDA in 2021. We're going to kind of talk a little bit about that a little bit later in the presentation. Everywhere else outside of the US is accepting ISO methods. With ISO methods, you're getting the same types of claims, your static SPF, your critical wavelength, or your water resistance. They also have a method specific for UVA protection, which is the ISO 24442 in vivo method. This method is uncommon to see in the US, but there are many markets outside of the US that require this test, in addition to the static or the critical wavelength determinations. So the first thing I wanted to talk about is the subject demographic criteria for sunscreen testing. We are going to focus specifically on static or just your general label claim determination. We'll touch on some other items, but the general idea of the test is the same with a few additions. In sunscreen testing, we are specifically looking for Fitzpatrick's types 1, 2, and 3. The descriptors of these Fitzpatrick types are just below, but we're looking at more of the pale to fair beige type of skin types. The reason for this, and specifically is that we are looking for people that might produce a sunburn. We would call that an erythema response. But you could see for Fitzpatrick's 1, 2, and 3, in their definition alone, these people are either always going to burn, often will burn, or will occasionally burn. On the other hand, we have the Fitzpatrick 4, 5, and 6 skin types. These skin types are not ideal for erythema responses, or what we're looking for in the determination of the sunscreen factor. According to the definition of these Fitzpatricks, you can see that they almost never burn or just do not burn at all. And again, we are looking for an erythema response. So these individuals are not ideal for our type of testing. The Fitzpatrick skin types are laid out in the regulations, in the protocols that we have to follow for both FDA and ISO in order to determine sun protection factor. This is a hot topic right now in the industry because we are looking for products that are more inclusive for all skin types. We have received a number of requests looking to do determination on Fitzpatrick's 4 through 6. Unfortunately, we are not able to do that in order to determine your SPF label claim. However, if you're looking for something more exploratory, something more research-based, it's definitely a conversation we can have in how we can help perform a test to get to your endpoints. Fitzpatrick skin types are specific to the FDA requirements. In ISO, they are looking for what's called an individual typology angle. This is a colorimetric evaluation of the skin in order to determine skin tone. For SPF determination testing, or ISO 24444, they are interested in enrolling individuals with an IT of greater than 28. These individuals would be considered to have intermediate, light, or very light skin. This is essentially equivalent to what the FDA is looking at in Fitzpatrick's 1, 2, or 3. It is a different method to determine the skin type. However, the idea is the same. These individuals are going to produce erythema responses. In addition to the skin tones that we're looking for evaluation, we also generally have certain criteria of all individuals for these types of tests. We're not looking to enroll people who are taking medications that are known to alter their responses to UV radiation, either to worsen those responses or to diminish them. We do not want to enroll people who have history of sensitivities to topical products. We are looking for individuals with uniform skin tone across the test area, so no pigmentation differences, sunburns, anything like that. So all of these things are taken into account when we enroll individuals into sunscreen determination testing. So in general, we have a schedule of events that are going to occur when you are testing your product with us. Step 1 is we're going to do a screening. So this screening will be either 3 or 5 subjects, and this is just going to give us a general idea of how your product is performing without having to go the full mile of doing the entire test. With these 3 or 5 subjects, we'll see if the product is protecting at your expected level, if maybe there's some formulation changes that we need to make, or even if your product is protecting at an even better level than you were expecting. These screening results will be provided to you, we'll talk about them, and we can determine the next best course of action. After our screening, if everything is looking great, we want to complete this 10-subject panel in order to obtain a label claim. We're going to go ahead and get that started. We're going to complete the study panel with a minimum of 10 subjects. Both FDA and ISO require a minimum of 10 subjects in order to put a label claim on your product. While we're finishing up that panel, In that meantime, we would recommend you do any other types of tests that you're looking to conduct in order to put on your label. We recommend during that time probably doing your UVA or your broad spectrum evaluation. At this point in the process, you already know that your product is passing towards your expected SPF value. So now you take that next step and just make sure it's meeting those broad spectrum requirements that you're looking to make. Once the 10 subjects are complete, you have your broad spectrum requirements done, your product is perfect. We're going to give you those results. We're going to put it into a nice report for you. We're going to give you those results within about 3 weeks following the end of the panel in that report. Prior to that, we're going to shoot you an email and let you know how your study wound up ending. Our goal is really to let you know as soon as possible how your product performed, what that label claim is going to be, and then we're going to follow with a written document of final report. Currently, our static and water resistance studies are taking about 4 to 5 weeks from the time we get your sample until the subjects are complete. And the in vitro or those broad spectrum UVA claim studies are taking about 2 weeks from start to finish. So we're going to go into a little bit about the study procedure. There are minor differences between the study procedures with FDA and ISO. However, generally speaking, the process is the same. The outcomes are mostly the same. There's some minor tweaks in the testing process, but nothing significant enough that's going to say your FDA results are going to be wildly different than your ISO results. Generally speaking, those results should be the same, and these minor differences in the process should not impact that. Okay. So first thing we're going to do is we're going to demarcate the sites and apply your product. After that, we're doing some UV exposures or irradiation. Thirdly, we're going to read the erythema responses after that UV irradiation, and then we're going to calculate the SPF. So to get us started, the demarcation of sites and product application. For your test, we are going to need a minimum of three test sites. These areas are 30 square centimeters, and within these sites, we are going to apply the investigational product or your test sample. We're going to have a control site, which is a reference sunscreen, and then we are going to have an unprotected site. So this site is not going to have any sort of product applied to it. The reference sunscreen is used as a control, and it's going to allow us to determine that the test was valid and that the results from that test are good to go. So the demarcation and product application, when we get your product, we are going to shake and stir your product. And we are very good shakers and stirrers. So we are there to make sure that your product, it has a uniform dispersion, it's homogeneous before we apply it to the test subject. We're going to apply uniform drops of the product using either a syringe or a pipette, and we're going to employ a weight by loss method to make sure that we are applying an appropriate amount of sunscreen to the site. The product is deposited at 2 milligrams per centimeter squared. So our sites are 30 square centimeters. So in total, we're applying 60 milligrams of sunscreen to the test site. So after that, we're spreading the product. We are spreading the product over the entirety of the test site. First, we're going to use circular motions, and that's going to gather all of those droplets together. Then we are going to use horizontal and vertical directions and using light pressure. And by the end of that procedure, we should have a very uniform and homogeneous spread of the product over the entirety of the test site. While we're spreading, we're making sure that our finger does not come off of contact with that skin. This is to avoid any deposits of product in certain areas. So again, it's just another step that we take to make sure that spread is homogeneous and even. After that spread, now we're going to make sure that it's acceptable. So we are going to take a UVA Woods lamp, and we are going to use that to visualize the uniformity of the application. What we're doing is we are looking to make sure that the product is spread evenly in that test site. It's from edge to edge. We're looking to make sure that that product didn't go outside of that test site. We're looking for streaking, clumping, anything abnormal to make sure, again, that we have a homogeneous layer of product over that site. After that, the product is going to dry for 15 to 30 minutes. And so the 15 to 30 minutes is a regulated amount of time that the product has to dry. During that time, our participants are lying still. We are not having a lot of movement, anything really to disturb the product that we had applied to the skin. If for some reason we pull out that Woods lamp and we're looking at the site and it's just not looking so good, there's some streaking, maybe there was a product that went out of the test site, we're not going to use it. It's not what we would consider an acceptable application. In that case, what we would do is this test site would just be voided. If another site on the back is available, we will attempt a new application and make sure that that next application is homogeneous. Next is UV exposure. We are using a multi-port solar simulator to irradiate over the sunscreen product. We're also going to be using that to irradiate on the reference sunscreen and on the untreated, unprotected site. These machines have to meet regularly validated and calibrated quality standards. The machines are regulated by the FDA and these international testing methods. So they have to meet specific UV requirements, quality standards, the beam uniformity standards. So everything that we are doing with these machines is to ensure the quality of UV exposure and therefore the quality of the results of test. So the doses that we're going to give over on top of these sunscreen products are really dependent on the expected SPF value of the product. For an example, if your product is an SPF 50, the UV doses that we give are going to be higher than if we were to be exposing a product of SPF 15. The expectation is that a 50 is intended to protect better than a 15. You'll see on the machine there are 6 ports. Each of these ports, each of these guides are independent of one another. So we can adjust each port to give a specific dose different than the others. So what we are doing on the product is we are giving progressively higher doses at each port. So what we're going to do is we're going to start with the lowest dose, and each one is going to get progressively higher throughout each port. These progressions are determined by the standards, FDA and international standards. For international, each progression could be 12 or 15%. For FDA, it's more dependent on the expected SPF of the product. So it would be either 15, 20, or 25% progressions. Step 3. So after we give these UV doses, we're going to give them on all the sites, the unprotected, the reference sunscreen, and the test product. We're going to send the participant home with some very specific instructions of things to do or not to do. And we're going to have them come back 16 to 24 hours after UV exposure. This time period is intended to allow those erythema or those sunburns to happen, okay? So, excuse me. So when they come back, we are going to evaluate each test site for what we call an MED. The MED is, by definition, the lowest erythemal effective radiant exposure that produces the first perceptible, unambiguous erythema with defined borders appearing over more than 50% of UV exposure subsite 16 to 24 hours after UV exposure. It's a lot of words, but there's really some key points within that definition that drive where we determine an MED to be. Okay, so those key words, we're looking at first perceptible. So the very first site in which we see erythema or redness or that sunburn. Next keyword is unambiguous. There's no ambiguity that that is the first perceptible site. It has to have defined borders. You can see by this example and by the multi-port solar simulator photo that our exposure ports are squares, right? So it gives us a very good idea of what a defined border should look like. And then that exposure, that erythema has to be more than 50% of the UV exposure subsite. So anything less than that is not going to be considered an MED. In the case of this particular example here, we can see that site 3 is what we would consider our MED, our first perceptible, unambiguous erythema with defined borders, more than 50% of the UV exposure subsite. After that, we're calculating the SPF. What we're going to do here today is we're going to do a little example, a little math exercise, because why not do that on a Tuesday? So let's pretend that we gave these exposures here for the untreated site. Again, reminder, the untreated site is unprotected. There is no sunscreen product on top of this site, so there's no protection from UV exposure besides your skin. These doses that we're going to give are much lower than you could see on the product protected site. Again, not protected skin here. So we're not giving some crazy UV doses that are really going to burn or harm the participants. In the middle there, you see on subsite 3 that we have what we call a midpoint dose. This midpoint dose is what we would anticipate the MED to be. So we are expecting that on this participant, this dose will be considered our MED, or that first perceptible, unambiguous, defined border erythema response. This midpoint dose is an expectation based on their skin type. So either the Fitzpatrick 1, 2, 3 or that ITA. This midpoint dose can be adjusted higher or lower dependent on the skin type. So if someone is more fair, we are going to give a lower midpoint dose, again, just to avoid burning them. If their skin is a bit more pigmented, we might raise that dose a little bit just to ensure we will have some sort of MED or some sort of erythema response on their skin. For the purpose of this example, we are doing 12% progressions. So we always start with our midpoint dose, and then subsite 4 will be 12% higher than subsite 4. On the other side of things, subsite 2 is going to be 12% less than 3, and then 1 is 12% less than subsite 2. Then for the product protected site, we are going to pretend for this example that we are testing an SPF 15 or an expected SPF 15 product. The midpoint dose also applies to the product protected site, but in order for us to get that dose, we are taking the mid dose of our untreated site, and we're going to multiply it by the expected SPF of this test sample. For this example, our midpoint dose on untreated is 232 joules per meter squared of effective energy. We're going to multiply that by 15, or the expected SPF, and then we get 3,408 joules per meter squared of effective energy. And then the same rule applies. Our progression's higher and lower. So for this example, we're still using that 12% progression higher and lower. So we sent this participant home, they come back 16 to 24 hours later, and now we are evaluating the MED. Our MEDs are always evaluated in the same position that they were irradiated in. For us at ALS, we irradiate and apply product in the laying down position or prone position. So all these sites are on their back. So they will come in, we will have them lay down on their back again, they will acclimate, and we will then evaluate the MED. The MED is evaluated by trained graders. They are all tested for visual acuity and color. We need them to be able to see those defined borders and redness or erythema. For this example, this person came back, their untreated site, their MED, or their first unambiguous perceptible erythema response with defined borders, more than 50% of the exposure site, was at subsite 3. At this site, we gave 232 joules per meter squared of effective energy. Then we went on to evaluate the product protected site, and they also had an MED at subsite 3 on this site. Now, in order to calculate the SPF, it's a very simple equation. The SPF is equal to the energy used on the MED of the protected site divided by the MED of the unprotected site. Okay. So for this particular example, our SPF of this individual is 3,408 joules per meter squared divided by 232, and their individual SPF of this product is SPF 15. Perfect. But not always. There is an expectation of variability in SPF testing. And this is kind of a quick example of why or how you would see that variability. It's not always so perfect that the MED or the protected site falls on subsite 3 and they're the same and they give you that exact SPF value you're looking for. So for these examples, if you were to pretend that the MED fell on subsite 4, which is 260 joules per meter squared of effective energy, but we still have the same scenario of the product protected site, you can now see that that individual's results are an SPF of 13.4. On the other side, if that MED had shown at 207 or that subsite 2, that's going to give you an individual result of SPF 16.8. Variability is expected in sunscreen testing, okay? To be honest, if I didn't see variability, I'd have a couple of concerns. And these results, these calculations of SPF are expected to take into account this variability, okay? So there are scenarios where the results would be rejected. Okay. The first of these is that the exposure series elicits an erythema response on all subsites. As you can see from the image, the erythema response is very clear, very defined on all of the 5 exposures that we gave, even from the lowest UV dose all the way to the highest. In this scenario, we're finding that the product is not protecting at the expected value, the expected SPF. So what would we do in this situation? 2 options. We would either recommend you lower your expected SPF. So if you were initially trying to claim an SPF 50, depending on the results, and this would be a conversation, we might recommend lowering it to an SPF 30. The other option is to reformulate the product. In this situation, we would stop testing at that point. You would only be responsible in terms of cost for the individuals we have completed up to that point. And you can go ahead back to the lab, reformulate, and send in a new sample for testing. With those new samples, again, we would go back to starting with screening results and then continuing on from there depending on the results. The next invalid or rejected result would be that the exposure series fails to elicit an erythema response on any of the subsites. So even at subsite 1 all the way to 6, we start low UV and get to really high. We're still not seeing an erythema response 16 to 24 hours later. This is a good problem to have. So this is telling us that the product is protecting even better than we had anticipated. In this situation, we would contact you and our recommendation would be to actually increase the target SPF. So if you came in expecting an SPF 30, it's likely you're looking more at maybe a 45 or a 50. So that tends to be a good problem to have. The last rejected result here is that erythema responses within the series are randomly absent or they're illogical. So this would mean that if you look at the image here, we have exposure subsites 1 through 6, really nothing at 1, questionable, something happening at 2, there's a clear MED at subsite 3, and then that next subsite 4, which is supposed to have a higher UV dose than 3, has no erythema response. Again, 5, we're not seeing anything, and then 6, there we are back to erythema. So this response is not following the logical progression of the UV doses that we gave. The logical progression is that it started low and the erythema should get more intense as we get up to subsite 6. That's not happening here. In this case, this would be a rejected response. So in this case, there might be something going on with the formulation. Maybe there's not, the ingredients within the formulation are not homogeneous and they're not spreading properly. This could be due to technical issues. So again, this case, what we would do on our end is definitely test another person to ensure the homogeneity of the product, ensure those technical issues. If we're still finding issues, again, this is a conversation we would have with you and we would discuss the product and the testing further. There are other scenarios where results would be rejected. This includes that the test subject is just non-compliant. You know, maybe we send them home and then we tell them to come back 16 to 24 hours later and they come back at 26 hours. Fortunately, it's a rejected test. We can't use them. They are not following the instructions as outlined in the regulations. Additionally, we have what's considered a technical failure, and this is simply maybe a failure of equipment during the treatment phases. Maybe the wrong exposure dose was given or an incorrect application was done on the wrong site. In those cases, that would be considered a technical failure, and we would remove that individual and we would test somebody else. In addition to all of these testing capabilities here on the left, we do water resistance testing for all regulatory agencies. We do in vitro testing, as mentioned, for critical wavelength, including the UVA1 and UV ratio, ISO 24443, which is essentially their version of broad spectrum testing. And we also do UVA testing according to ISO 24442. This is an in vivo method. This particular method is going to give you that label claim that you see where it says PA+++. The water resistance testing essentially is an add-on to the testing procedure that we described. After the product is applied and it goes on their back and it dries for that 15 to 30 minutes, after that, we're going to do water immersions. Those water immersions are going to entail about 20 minutes in a bath or water immersion device, and then a certain rest period outside of the device. That rest period could be anywhere from 5 to 20 minutes, depending on the regulation we're following. And with that water resistance testing, you are going to be able to determine and label if the product is either 40 or 80-minute water resistant, or very water resistant, or just regular water resistant as they have in the standards. Additionally, we do some other photobiology tests here at ALS. Safety testing is one of our major ones. Everybody is concerned with the safety of their test products, I hope. So our safety testing is phototoxicity and photoallergy. So we will be able to determine if the product is having a phototoxic or photoallergic response after UV exposure. Furthermore, we do blue light, visible light, SPF on wet skin. We can evaluate the product in salt or chlorine water, ultraviolet protection factor of textile, so your UPF value, and photo stability. So I wanted to take some time to talk about some challenges that we see in sunscreen testing. Over my many years working in sunscreen testing, you come across a lot of challenges that brands and formulators will face once it gets to us in the lab. The one of utmost importance is the ability to spread the product. If the product does not, if we cannot spread it well, it will not test well. Okay? When your product gets to us, what we're going to do ahead of time, before we even start testing on people, is we're going to evaluate the product ourselves. We're going to take a little bit out, we're going to apply it, and we're going to see if that we can spread that product well in order to obtain uniform coverage. If we cannot, we are going to talk to you about it. We're going to talk to you about whether or not if the product is drying too quickly and that's affecting the spreadability. Is it clumping or sticking together? Or does it even separate too quickly like some sort of biphase products might? In the image here, the example, this particular product was pilling or clumping before we could even get it across the test site. In this case, we spoke with the client and we let them know what was happening. And for them, that was atypical. So there was something happening maybe in the batching process or the formulating process that they were not expecting. So it's super important to make sure that that spreadability of that product is decent, because again, that will impact the test. In this particular case, it impacted the study altogether. We decided that this was not an ideal product to test, and the brand took it back to the formulators. Formulation considerations are a huge challenge, especially today with sunscreen products. We always will recommend that you review these formulations and verify your product is what you're expecting before it even gets to us at the lab for testing. You should verify that the percent actives in the sample correlate to what is expected of the SPF value. We are finding very often that even though we put X% of active ingredients into the sample, that percentage does not necessarily correlate to an SPF 50 of what you're expecting. It's super important to understand what that correlation is from actives to SPF value. You should check the final formula. Actives are present in the expected amount in the formulation. With an assay, you can come and say, we are expecting there to be 25% zinc oxide in this product. And with that assay, we can let you know if yes, there is indeed about 25%, or maybe something happened in batching that now we're finding that there's only 15%. So you expected 25, but now it's 15. Do you need a pH verification of the formula? pH can impact the actives and how they're performing. Is the formula stable? And we'll talk a little bit about this a little bit further, but obviously stability of a sunscreen product is of utmost importance. Are there other ingredients in the formulation that may be hindering the effectiveness of the actives? So maybe we did verify that the percent of the actives in the sample correlate to what is expected of an SPF value. However, even though there's a certain percentage of actives, the entire formulation impacts how they perform. Okay, so it's important to understand, or your formulator to understand, how these ingredients interact with one another and how they may impact the effectiveness of the sun protection. You want to verify that the ingredients within the formulation are dispersed uniformly. We can only do so much on our end when the sample gets to us. You know, we shake, we stir. Our goal is to make sure that what we're applying to the back is homogeneous within the sample we receive. However, there might be some things going on more microscopically that we just cannot fix. We cannot aid in during the testing process. With zinc oxide in particular, it's really important that you verify that these ingredients are not agglomerating, they're not sticking together. Lastly, I think it's super important that you check where your ingredients are coming from. Not only are you responsible for checking your sunscreen to make sure that it's meeting all these standards, the ingredients, where you're sourcing these ingredients, they also have a responsibility to check those ingredients to make sure that they are up to standard so that you can then put them in your products. Ask questions. Do audits, see reports. We're super audit friendly here. It's very important that you're checking where you're getting your ingredients, where you're getting your testing done. Lastly, I wanted to talk with challenges is natural sunscreen ingredients. This is huge in the industry right now. Everybody wants to go natural. It's super important to do so. In addition to our other chemical sunscreens, which also act very effectively. However, what we're finding in the testing world is that zinc oxide is generally not a very effective filter. So formulating a sunscreen with high SPF value can be rather difficult with zinc oxide. From my experience, what we're seeing is that zinc oxide tends to have a 1-to-1 ratio of percent to SPF value alone, not within a formulation. So if you're putting about 10% zinc oxide in a product, we tend to see an SPF value of 10. Okay, zinc oxide is a less efficient filter that we're finding than titanium dioxide, but when you combine them, they can be quite effective as both a UVA and UVB sunscreen. And then lastly, more filter does not necessarily mean the product is more effective. As I had mentioned earlier, we have to look at the formulation as a whole and how that ingredient is interacting within the formulation. So we've seen that you will get a product with zinc oxide at 20%. The product is not meeting the target SPF value. We'll get a reformulated sample. Now it's at 25%, but we're still seeing the same results. So we have to consider what else is happening within that formulation. The challenge with that is more filter does not can lead to diminishing sensorial aspects like thick products, leaving a white cast. And that's also a big challenge we're seeing in sunscreen products in the industry right now. We want effective sunscreen, but we also want something that's nice to put on our skin, right? So it's a very challenging ingredient to work around in general, and it's challenge across the sunscreen board of making sure that all of these factors are working together. All right. What we recommend on our side of things is there are tools that can be used. There are sunscreen simulators that can be used to kind of help guide what these filters will represent in an actual SPF value. Just recently, I tried out 2 filters and I put in 25% zinc oxide and the resulting SPF was 10. So it's very interesting to see. And to be fair, these simulators are not law. Things can change. People add boosters, mixing the ingredients together. But they are a really nice guide to help understand how a product is going to perform during the formulation process. A main takeaway for me for this particular topic on zinc oxide is to always try to work with formulators who are expert with inorganic filters. These are hard things to work with. So find someone who just really knows what they're doing. So then we want to talk a little bit about regulatory considerations. When you have your sunscreen product, these are regulated products. So it's important that you understand what you have to do in terms of labeling, what ingredients are acceptable. So for labeling, you want to understand what is the accepted SPF value based on your study results that is allowed where you're marketing. SPF label values in the US, for example, currently can be up to 100. We see those on the market right now. But in other countries in Europe, there's a cap of like a 50 plus. There are certain criteria that you have to label your product with. You want to consider what terms, statements, warnings, and usage are required to be on the label. The FDA specifically has very specific wording that you have to use. For example, if your product is water resistant or about skin cancer concerns. You want to ensure proper labeling of the SPF value, broad spectrum, and or water resistance statements. There are even specific criteria of how you can present those on your label, including even as far as how big they have to be on certain labels. In terms of ingredients in the formulation process, are you using approved ingredients at the accepted levels? We've been asked to test products for sunscreen protection who are not using any approved ingredients. So we have to have a conversation with these people and say, this is not a marketable product because you have to use specific ingredients in there. Are you considering ingredients that are generally recognized as safe and effective? Right. So again, these products, there are ingredients that are approved, but some that are considered safe and effective and some that are just not. So we'll talk a little bit about the FDA proposed rule. So right now, there is an FDA monograph that's published in 1990. There is an FDA labeling and effectiveness rule published in 2011. And now there is an FDA proposed rule, most recently here in 2021. This proposed rule is expected to go to a monograph, ideally, but as many of you I'm sure know, the FDA just takes their time. So with this proposed rule, we are expecting there to now be a maximum label SPF in the US. And that maximum label SPF is going to be 60 plus. So there should no longer be products on the market that are saying they are SPF 100, right? The maximum label SPF value, according to international regulations, is 50 plus. So the FDA is kind of coming on board with these international regulations with a minor tweak. Zinc oxide and titanium dioxide will be two ingredients that are generally recognized as safe and effective. And these will be the only two ingredients out of all the ingredients listed in the monograph for sunscreen products that are generally recognized as safe and effective. Two ingredients will not be safe and effective, and all other ingredients are still considered not safe and effective due to inefficient data to determine them as safe and effective. That does not mean that they cannot be used. It means that essentially use with caution. Maybe down the road, these might be generally recognized as safe and effective or not, but the FDA just doesn't have enough information on them to determine either way. The two ingredients that will not be generally recognized as safe and effective should not be used in sunscreen products. And they will also have a new broad spectrum requirement for products with SPF greater than or equal to 15. So currently, the requirement to label a product broad spectrum is just a critical wavelength of 370 nanometers or greater than 370 nanometers. This new the proposed rule is also stating that not only should the requirement be greater than or equal to 370 nanometers, it should also have a UVA 1 over UV ratio greater than or equal to 0.7. So what the FDA is trying to do here is to increase the UVA protection requirements. UVA is the leading cause of skin cancer, while UVB is the leading cause of sunburns. So what they're trying to do is make all products have a requirement of having this broad spectrum protection. And again, this is really just to aid in consumer safety. In addition, some other points that are proposed in this rule are new label requirements to help consumers read and understand the product more easily. Spray sunscreens are going to have an additional testing requirement to make sure that we're reducing inhalation harm. And they are completely getting rid of insect repellent plus sunscreen products. So a product that is an insect repellent cannot be a sunscreen, vice versa. And then I wanted to touch on MoCRA, the Modernization of Cosmetics Regulation Act. So this act is intended to strengthen the oversight the FDA has on cosmetic products by requiring and establishing pre-market review and reporting requirements. Generally speaking, MoCRA is about safety and quality. Okay? So this act is an additive to everything else that's currently enforced in the regulations. So all these sunscreen regulations that we're currently following still got to do it. This is an addition to everything that we're doing now. Okay? So currently, everyone should be preparing for MoCRA. It is intended to go into effect a lot quicker than we're all expecting, or not expecting, I should say, but quicker than we are probably ready for. So in this act, we are looking at safety substantiation, mandatory allergen labeling, facility registration, GMP regulations, adverse event reporting, and recall authority, to name a few things. All of this, like I said, is to aid in the quality and consumer protection. With quality, it's important to also understand that your testing facilities and your sourcing is following quality regulations as well. So for us at ALS in our sunscreen testing, what type of quality are we doing, right? Are we doing these solar simulator checks to make sure that the equipment is functioning properly? Are we evaluating these sunscreen products under these UV woods lamps to ensure the quality of the application? Furthermore, us at ALS in the sunscreen department, we participate in it's called BIPIA. It is a ring test essentially, and we participate with a number of labs. And what we're doing with BIPIA is we're ensuring the quality of our application and our erythema assessments to make sure that we are upheld to the quality of other labs. So not only is it a quality, but it's an interlaboratory comparison to make sure that we are doing everything that we can to ensure our application, our erythema readings, our testing procedures are up to standards. It's, again, to touch on that quality and especially the GMP regulations. You're looking for formulators and testing facilities who are following GCP, GMP regulations. Very, very recently, probably within the last month, if you see, there's some FDA warning letters going out, and these are specific to GMP violations. One more recently was specifically targeting a sunscreen product. So MoCRA is really starting to gear up. That oversight is really starting to happen, and it's only going to become more prevalent. So it's very important that you as a company, as a brand, and us as a testing facility are really up to high standards. And to add, I know you saw a very nice QR code on that. ALS does offer a service to help you understand if your company, your products, your brands are up to what MoCRA would consider like their standards. So we are also here to help you to make sure that when this act truly goes into effect, that it will be a seamless transition for you. At ALS, we're here. We have a variety of services to offer, and our goal really is just to make it simple for you and your testing of your products. We offer comprehensive testing that will take you from formulation to market. Okay. In addition to that effectiveness testing that we're doing, that SPF labeling requirements, a lot of products are also intended to have anti-aging or effectiveness, other effectiveness properties. With that, we can also help you with those anti-aging, those claim substantiation types of tests. We are a very prominent lab for safety testing. So our human repeat insult patch test, it's a very prominent test, something that we highly recommend you do for your product. So in addition to other safety testings of sunscreen specifically, like phototoxicity and photoallergy. With formulations, we can talk about your preservative effectiveness testing, your stability testing, assays, impurities, toxicology. So again, we're taking you from start to finish. Something specific about our HRIPT test is that we are running them weekly. So it's very beneficial that we can turn around these types of tests in a shorter amount of time, maybe in comparison to some other laboratories that are also doing this type of test. For all of our types of tests that we can do, check out our website. Make sure specifically to go to the personal care and OTC testing capabilities section, but it lists out everything very nicely. But yeah, so again, we are start to finish one-stop shop. So that is my webinar for today. I'm really grateful that everybody took the time out of their day to come and listen to me talk about something that I love to do so much. And again, if you have any questions, you can always reach out to me. You can reach out to our full team here at the beauty.usa email address. But again, our goal with ALS and your products is we want to make it easy. We're here to make it easy for your testing. So thanks again, everyone. And then I guess I can open it to if anybody has any questions that they would like to ask. I know there's a Q&A in the Zoom. You are all going to have to bear with me because I'm probably not good at it, but I'm happy to answer any questions you might have. All right. Looking like we're good to go. Everyone enjoy the rest of your day. Thank you so much. It was really great having you here, and I'm sure we'll talk soon.